In the late 1800s, scientists tried to understand the brain by drawing maps of what they could see. Under early microscopes, nerve tissue looked like a tangle, and the big prize was finding the true working parts. Neurons became the stars. Their long extensions suggested wiring, direction, and communication. Around them sat a quieter material that seemed to hold everything together. It received a simple label, glia, meaning glue. That label carried a powerful assumption. If neurons carried messages, then glia just filled space. Many anatomists treated it like packing material between the important cells, a kind of biological mortar. They measured neuron shapes, traced pathways, and argued over connections, while glia stayed in the background. No one framed it as active tissue. It did not look like a system that cleaned, digested, or protected. It looked like leftovers. So early brain science moved forward with a blind spot built in. Experiments and theories focused on electrical signals and neural circuits, because that was what seemed to explain behavior. The surrounding cells were rarely given questions to answer. If waste built up, if damage needed repair, if the brain required constant care, that work went unimagined. Glia were present everywhere, yet cast as passive. The brain's hidden maintenance crew went unnamed. In the early 1900s, the picture of the brain did not just sharpen, it changed. New stains and better microscopes made the so-called filler stand out in ways no one expected. Instead of one bland background, there were different shapes, different patterns, different boundaries. Some cells stretched out in star-like webs. Others looked smaller and more compact, scattered like watchful dots. Those differences mattered because they suggested identity. The star-shaped ones became known as astrocytes. The smaller, tougher-looking ones became known as microglia. Names were not just labels here, they were a shift in thinking. If these cells came in distinct types, then the brain's glue probably did not do one single job. It probably worked in a division of labor. That idea quietly challenged the old hierarchy. Neurons still held the spotlight, but the supporting cast no longer looked passive. Different glial cells sat in different places, wrapped around different structures, and appeared built for different tasks. The evidence did not yet spell out what those tasks were, but it made a new question unavoidable. If glia were specialized, what exactly had they been doing all along? By the mid-1900s, the question stopped being what microglia looked like and became what they did. Researchers kept brain tissue alive in the lab and watched these small cells change shape and move. They did not sit quietly in the background. They reached outward, tested their surroundings, and gathered loose material around them. A support cell had started to act like an active worker. Then the key behavior became impossible to ignore. When scientists added damaged cells or bits of foreign material, microglia pulled the pieces in and broke them down. This was phagocytosis, the same basic trick used by immune cells in the rest of the body to eat threats and trash. The surprise was not that it happened. The surprise was where it happened, inside the brain itself. That result changed the mental map of the brain. It meant the brain did not rely only on outside defenses or slow, passive decay to handle mess. It carried its own resident cleanup crew ready to swallow debris and respond to pathogens. Microglia were not just another kind of glue. They acted as dedicated maintenance cells with a built-in appetite for what did not belong. By the late 1900s, the spotlight shifted to astrocytes, the star-shaped glial cells wrapped around synapses. Synapses had looked like simple handoffs. One neuron released a chemical message, the next one received it. But experiments kept showing something didn't add up. Signals lasted too long or spread too far unless something else stepped in. Astrocytes did. They sat right beside the action, positioned to control what the neurons left behind. Researchers found that after neurons released neurotransmitters, astrocytes helped clear the leftovers. They pulled these chemicals out of the gap, reducing stray messages that could confuse nearby synapses. That changed the basic picture of brain communication. A synapse was not just two neurons talking. It was a shared space, managed minute by minute. When astrocytes failed to clean up fast enough, the chemistry lingered and the message risked turning into noise. Astrocytes also buffered ions, especially potassium, which built up when neurons fired repeatedly. Without control, that ion buildup shifted the electrical balance and made neurons misfire or become unstable. Astrocytes soaked up the excess and redistributed it, keeping the local environment workable. The consequence landed hard. Glia did not merely support the brain's wiring, they ran the chemical housekeeping that let synapses function safely, second by second. By the 1990s, a new suspicion took hold. Glia did not just support thought, they helped shape it. Experiments kept showing the same pattern. When astrocytes became active near synapses, neurons changed how strongly they talked to each other. This mattered because learning depended on those changes. The old picture had neurons as the only decision makers. Now the supporting cast started to look like part of the script. Researchers called one proposed mechanism gliotransmission. The idea sounded almost wrong at first. Glial cells released chemical signals that influenced nearby synapses, nudging whether connections strengthened or weakened. Results varied between labs, and the debate stayed lively, but the direction of travel stayed clear. Glia repeatedly appeared at the exact moments when circuits adjusted. Plasticity no longer looked like a purely neuronal event. It looked like teamwork. At the same time, synaptic pruning sharpened the point. During development and experience, the brain removed some connections and kept others, like trimming a crowded tree. Microglia emerged as key players, inspecting synapses and helping eliminate the ones that did not fit the network's needs. That meant learning involved subtraction as well as addition. And it meant cognition depended, in part, on cells that earlier generations barely noticed. In the early 2010s, a stubborn question finally got a fresh answer. The brain produced waste like any hard-working organ, but it lacked the obvious plumbing the rest of the body used. Then, researchers proposed a hidden route, a clearance system that moved fluid through the brain itself, washing out byproducts as it went. They called it the glymphatic system, linking glial cells to a lymph-like cleanup job. The idea sounded simple, but the mechanism was bold. Cerebrospinal fluid, the clear liquid that bathed the brain, did not just sit in place. It flowed inward alongside blood vessels, then pushed into the tissue through channels formed by astrocyte end feet wrapped around those vessels. That movement helped mix fresh fluid in and pull used fluid out. Waste products hitched a ride in that stream. What changed everything was that this pathway did not stay theoretical. Imaging work tracked fluorescent tracers moving along those vascular corridors and spreading through the brain in real time, then draining away. It looked like a system, not a leak. A brain-wide rinse cycle suddenly seemed possible, and astrocytes sat at the center of it, shaping where fluid went and how efficiently waste could leave. Follow-up tests pushed the glymphatic idea past a one-time discovery. Researchers measured how quickly waste markers moved out of brain tissue, then compared the same process across different states. The results surprised them. When the brain stayed awake, clearance plodded along. When it fell asleep, the pace changed. It did not just continue. It shifted into a higher gear, as if the system had been waiting. They repeated the comparisons again and again, tightening the controls. Sleep kept showing the same pattern, more fluid movement through the clearance pathways and faster removal of what did not belong. It turned sleep into something measurable, not just restful. It looked like a scheduled maintenance window. A real cleaning shift happened, and it happened reliably enough to track, quantify, and predict. The experiments also tied that shift to glial work. The flow followed routes shaped and supported by astrocytes, and the whole process rose and fell with glial function. That connection mattered. It meant sleep was not only a pause for neurons, it was a time when support cells took the lead, moving fluid, clearing waste, and resetting conditions for the next day's activity. Once researchers accepted that the brain ran a real waste removal shift, the next question followed fast. What happened when that shift slowed down? Studies in aging animals and human tissue pointed to the same pattern. The flow that carried waste away became less efficient. The cleanup cells worked harder, but they also became stressed. Small leftovers that once got cleared routinely began to linger. Those leftovers were not harmless. Some were sticky proteins the brain normally made and then removed. When clearance lagged, these proteins piled up in the spaces between cells, and damaged parts of neurons stayed in circulation longer than they should. At the same time, microglia often stayed switched on, sending inflammatory signals as if danger never ended. Inflammation stopped being a short response and became a chronic state. That combination changed how scientists thought about long-term risk. Slower clearance meant more debris. More debris kept immune cells activated, and constant activation risked collateral damage, stressing synapses and altering normal repair. Over years, not days, this feedback loop looked like a pathway toward neurodegeneration. The idea landed with force. When the brain's cleanup system fell behind, toxic buildup and inflammation could quietly compound over time. By this point, glia no longer looked like background support. They acted like a working organ, and the easiest way to grasp it was a surprising metaphor, a second stomach built into the brain itself. Microglia handled the rough work. They moved in on worn-out cell parts, damaged connections, and leftover protein clumps, then swallowed them and broke them down. It was not just cleaning, it was digestion happening quietly in living tissue. But digestion needed supply lines. Astrocytes provided them. They sat between blood vessels and neurons and controlled what got delivered and when. They helped shuttle fuel from the bloodstream, kept the chemical environment stable, and managed the timing so neurons could keep firing without crashing. At the same time, astrocytes shaped the movement of fluid through brain tissue, guiding wash-like flows that carried dissolved waste away from crowded spaces. That combination changed the frame. Brain health did not depend on electrical signals alone, or even on cleaning alone. It depended on metabolism and cleanup acting as one system. When energy delivery stayed steady, cells ran smoothly and produced less damaging spillover. When fluid movement stayed organized, waste cleared before it piled up. Microglia and astrocytes linked these steps together. The brain fed itself and it cleared itself using the same supporting cast. By the present day, glia no longer sat in the background. They became one of the busiest frontiers in brain science. Researchers tracked how astrocytes controlled fluid routes, how microglia shifted between repair and harm, and how these choices changed with age, stress, and disease. The old assumption of passive filler collapsed under data. Caretaking turned into a measurable job, with steps that could be watched, quantified and potentially tuned. Sleep-linked waste removal became a practical target, not just a curiosity. Teams measured how well the brain cleared waste by following fluid movement and chemical traces, then compared good sleepers to poor sleepers, younger brains to older ones. Clearance patterns started to look like early warning signs. If the cleanup slowed, risk could rise before symptoms appeared. That shifted the goal from reacting late to spotting trouble early. New therapies aimed at the caretakers themselves. Some approaches tried to calm chronic microglial inflammation, so cleanup did not turn into collateral damage. Others focused on astrocyte channels and blood vessel coupling to support nighttime flow and waste transport. Even sleep habits entered the medical conversation as biology, not willpower. In the end, today's tools treated glia as living infrastructure, cells that decided whether the brain stayed clean.